please put a hand together for Dr. Doss.
All right, can everyone hear me? Or is that coming through? Yes? Yeah? Okay, cool. So yeah, I originally was going to be talking about the effects of various drugs on episodic memory, but then it turns out the last participant for the study ended up being a writer for Vice and ended up putting this article out. So I kind of felt a little bit of pressure to get this information out as soon as possible. So sorry if some of these analyses are a bit half-cooked. I was literally still writing analyses like a couple days ago. But I think that we can get a little bit of a picture of what Salvinorin A is doing to the brain. So the first thing I was going to do was show you a video for those of you who don't know what salvia is. And I think I can do it through here now. So.
Hello, and welcome to Driving on Salvia. Today we're going to take the car out into the streets, and we're going to do some K-turns, and then we're going to do some U-turns, and then we're going to go ahead and parallel park in a spot smaller than we have before. To start it all off, however, we need to take a hit of our salvia. Once you have that done, you're going to hold it in for about 20 seconds, or until your vision begins to vibrate. And you'll be good to go. Check your mirrors. Make sure that they're pointing in the right direction.
So you can see it's already coming on pretty strong.
I'll be back in a moment.
So he held that in for about 40 seconds, which is about what we do actually in the lab. Typical reference. So you can see he's getting some kind of visuals. So unfortunately, seeing cats is not a common symptom of salvia, but you know. So what's kind of interesting about salvia is that sometimes people will forget altogether that they even used it, and I think right now he's kind of clicking maybe back in and realizing where he is and what's going on. Okay, so if you can't tell, it's kind of difficult to study a drug like this whenever it has, you know, especially when we're trying to keep people from moving in a scanner, and clearly there's a lot of effects that make people want to move. So normally I have little like transitions between points, so I don't just load you up with too much text, but whatever, we'll just deal with this. So salvinorin A is the main psychoactive constituent of Salvia divinorum. So that's Salvia divinorum over there. This was only discovered in about like the 90s, so it's relatively recently that we've kind of figured out the main action of Salvia divinorum. And then traditionally, salvia was used by native Mexicans who used it kind of sublingually, and then it was rediscovered by Albert Hofmann and Gordon Wasson, and then it was culturally appropriated by us, and now we smoke it, and it has a super intense effect. So it has a super it's sharp come up, but there's also shorter lasting effects. As you could see over there, he was probably starting to sober up a few minutes in. And what's weird about salvia is that it's still actually legal in 21 different states, including in Maryland, but I don't think here in Pennsylvania, so come see us and get some salvia. So what is it? So some people will classify it as a dissociative hallucinogen, and what's really strange is there's kind of a wide range of effects even within individuals that can be everything from euphoric to terrifying. And as far as comparability to other drugs, there's an unpublished study that's actually still on a preprint server, but done by scientists, in which they used the trip reports from Erowid.com, and what they actually found was that various drugs clustered together in terms of when they used a language classifier, such as psilocybin and LSD, but weirdly enough, DMT and salvia kind of cluster together. And now, I don't think that these two drugs are similar. I think anyone who's probably done both of the drugs wouldn't necessarily say that they are, but I think the mode of administration is one reason why. If you take anything that's super potent and rapid acting, you're going to use words like transportive and otherworldly, and so because of that, I think that's one reason why they overlap. But there is some subjective overlap as well. So some people will report that DMT can result in like near-death experiences. I mean, David Nutt just actually put a study out not too long ago in which they used I think a survey, like a 15-item survey from the 80s, and there was more near-death experiences from DMT. But again, some people recently just used the trip reports from Erowid.com, and they compared them to actual near-death experience reports, and they actually found that ketamine came in first, then followed by salvia, and then serotonergic psychedelics. But another similarity is also that as David Nutt was talking about, was that people will kind of have these illusory social interactions, or entity experiences, if you will. And so that's another kind of interesting thing that a lot of people will report from salvia as well as DMT. And so what does salvia do? So unlike DMT or other classic psychedelics, it doesn't modulate the serotonergic system, at least not in the way that classic psychedelics do, but rather it binds to the kappa opioid receptor. And so this is not the same receptor as drugs like heroin or opium or anything like that. This is a totally different receptor, which has a different endogenous ligand than those other receptors. And as David Nutt mentioned, there's a high density of them in the colostrum. However, here's a PET image, and you guys see that they're distributed all over the brain, particularly densely in the prefrontal cortex as well as the temporal lobe. And salvia is not necessarily special. There's other kappa opioid agonists, as somebody asked about. Ibogaine is actually a kappa opioid agonist, as well as it also binds, I think, to the serotonin 2A receptor. And then there's a drug that was at one point attempted to be put on the market for pain treatment, and it didn't make it too far in clinical trials because, as this report says, it can cause some kind of psychotic-like effects. So what they reported was that he became agitated, began swearing, mistrusting others, and accused staff of seeking to harm him. Afterward, the subject reported that he had seen and felt waves. So I think it's certainly, that sounds pretty psychedelic, maybe bad trip initially and then something better later. So salvinorin A is not special, is just the point I wanted to make, but in terms of binding to this receptor. But this receptor does seem to have some kind of psychedelic-like effects. So why should we study salvinorin A? So I think I think one of the most obvious reasons is to better understand what the kappa opioid system is doing, and that this might actually be important for understanding something like psychosis. So people have used ketamine, it's an NMDA antagonist, as well as now classic psychedelics to kind of model different aspects of psychosis, and this might be modeling other aspects of psychosis. Less known is that there's at least been some animal literature showing that it can have kind of rapid antidepressant or anxiolytic effects, and there's at least one one case report in which somebody said that they self-medicated with salvia. There's also been some research showing that it can be anti-addictive, and I think ibogaine kind of speaks to that. And then lastly, this has been, I think this has been an empty promise in a lot of psychedelic research, is that people really keep talking about how these drugs will tell us something about consciousness or cognition more broadly. But I mean, I was just talking to David Nutt, and I have yet to see a single study that's actually told me anything that I didn't already know about some underlying principle in the mind or brain, but I'm open to that happening. Okay, so there's been a lot of work with Salvinorin A in behavioral, in animal models, and so first I'm just going to go over a few effects in behavior, and then we can go over some of the brain effects, and then we'll talk about what it does in humans. So first of all, rodents do discriminate between Salvinorin A compared to LSD or ketamine. So a lot of times people will use certain behavioral assays to see whether or not two drugs are similar. So for example, if you take like LSD, a rodent might not be able to discriminate necessarily between like LSD and let's say psilocybin. However, in this case, they could discriminate between Salvinorin A compared to LSD or ketamine. It's been shown to impair episodic memory, so kind of like your memory for yesterday, what you ate for dinner or something, as opposed to working memory, which is holding information in mind. And these are, there's kind of like proxies to these sorts of processes in rodents, and it's been shown to impair episodic but not working memory. And then it was shown that lower doses can produce preference. So a rodent will go back to a side of a cage in which it receives low doses of Salvinorin A because it probably wants more, but then at higher doses it actually will stay away from that side of the cage. And then this actually maps onto certain dopaminergic effects. So a lot of people think of dopamine as a reward molecule. It's a bit of a simplification, but you can see more dopaminergic release in a reward center in the brain with lower doses, then you can actually see suppression at higher doses. And then it's been shown to increase activity throughout the cortex, but then also decrease metabolic activity in subcortical regions. And then much like as David Nutt was showing with classic psychedelics, it's also been shown to have broadband decreases. At least kappa opioid agonists have been shown to have broadband decreases in oscillatory power. So in one of the first Salvinorin A studies, they gave, this was only 8 subjects, but they gave 16 different doses, so really low to really high. And what you can see here is that across these doses, that maybe peak effects happen at about 2 to 3 minutes, and that they kind of taper off by about 20 minutes. So a lot of people think that salvia is only like a 3 to 5 minute drug, and at least with most of the participants we ran, they were still feeling effects by about 15 minutes. And so in this first study, that what they found was that moderate to higher doses can lead to like hallucinogenic as well as like mystical effects. And then there was dose-dependent decreases in episodic memory, much like what was shown with the animal models. And then there was frequently reported illusory social events. And then in this other study, what they found was that compared to other kind of altered states, that participants reported salvia to being unique, although the most common comparison was dreaming. So people thought it was maybe more similar to dreaming as opposed to like an LSD experience. And then kind of lending credibility to this idea that maybe there's kind of longer lasting after like antidepressant effects, people found that, that at least most participants reported that there was some kind of positive effect over 24 hours later. That being said, I think there was still a small fraction of participants who actually said there was also negative effects lasting 24 hours later, so take that with a grain of salt. So although we're the first, I'm going to be showing you the first like fMRI data with Salvinorin A, there has been some other brain data. So with EEG, for example, which is like MEG as Dr. Nutt was talking about. And so in this study that was done at Yale, they used like 8 to 12 times higher doses than any other study, but they also used, I think, a vaporization method that was much less efficient. So I wouldn't ever recommend taking 8 milligrams or 12 milligrams of Salvinorin A. But in this study, what they found was that at least at 7 minutes in, when there are still some effects, that first of all, there was no impairments in working memory. So again, this kind of maps on to what was seen with animal models. And then they saw at least numerical decreases across all oscillatory bands, with the strongest effect being in beta and theta. And then in another study, what they actually did was they co-administered Salvinorin A with either a serotonin 2A antagonist or a kappa opioid antagonist, with the idea being that one of these will block all of Salvinorin A's effects, and that's what they found with the kappa opioid agonist, but not with the serotonin 2A antagonist. And so this would suggest then that most of Salvinorin A's effects are coming from the kappa opioid receptor. So before I go into what we did, where I'll talk to you a little bit about some of these resting state networks that was mentioned by David Nutt. And so the first, probably most studied network throughout the brain is the default mode network. Although it is pretty active at rest, it's actually involved during a lot of tasks beyond rest, such as when people are thinking about what someone else is thinking, when people are remembering the past, or if they're simulating the future. These are all situations in which this network seems to be involved. And it has been shown that this network has particularly strong connectivity in depression, but there's also other disorders such as ADHD in which you can see strong connectivity. And what has, as was shown, that classic psychedelics as well as ketamine can decrease connectivity within this network. So another network that people talk a lot about are like task-positive networks. These are kind of slightly ill-defined. There's many different ones, but at least one thing that they all have in common is that they seem to be that it seems to be involved with like external kind of attention. So whereas default mode network might be more of like an internally directed attention, this is more about what you're actually doing in the outside world. And so some people have shown that there's an anti-correlation with the default mode network and these task-positive networks, although that might be an artifact of the way you analyze your data. And then at least in one study done from David Nutt's lab, they found that psilocybin increased connectivity between these two networks, although they didn't replicate that with LSD, and it also wasn't shown by a separate group with DMT. And it's also not shown that these two networks don't become more correlated from ketamine either. So another network that people have talked a lot about is the salience network. So this seems to be an important network for like integrating or orienting towards emotional or sensory stimuli. And in the first paper, I think, with the salience network, they found that it actually also correlated with anxiety. And it seems to, what some people think is that it kind of mediates between like task-positive networks and the default mode network. And one study from David Nutt's group again found that LSD increased connectivity with the default mode network and the salience network. And this was also found with 2 different studies with ketamine. So I'm just kind of giving you an idea of what different networks, what we might see with something like Salvinorin A, which is a related but obviously different drug. And then lastly, there's a bunch of the visual cortex can be kind of broken up into multiple different networks, sometimes 2 or 3 different networks. And so what I'm showing you here is like a medial, a lateral, and then like a occipital polar visual network. And so in the LSD study, it was found that there was actually decreases in connectivity across all these 3 networks, and there was no differences in between network connectivity of these networks. However, they did this really cool study in which you can actually map visual space. So like a particular area of the back of your cortex is going to be associated with like this area of visual space, and then this area of visual space will be associated with a different part of your cortex. And this is kind of done in like a hierarchical fashion from a region known as V1 up to V3. And one thing they actually found was that you can get like a retinotopic. You can get increases in connectivity in this retinotopic fashion from LSD while people's eyes are closed. And so it was a really cool way of essentially showing that people see things. Oh, sorry, one thing I should mention is that that pattern of connectivity is seen when people are actually perceiving things versus just kind of, keeping their eyes closed. And so they showed in a really cool way essentially that, yeah, when you close your eyes, you see things on LSD. And then it was also shown with ketamine that you can that there's an increase in connectivity in a medial visual network, but not necessarily a lateral visual network. So both of these drugs seem to modulate visual cortex, but kind of in different ways, perhaps. So in the present study, we used a single-blind, placebo-controlled, non-counterbalanced design. When you see the device that we used, you'll see why we had to do it single-blind. It's really difficult to kind of mask to mask whether or not the researcher is blind to the Salvinorin A manipulation. And the reason we don't do things counterbalanced is the same thing, I think, as why David Nutt doesn't do it counterbalanced, is that we didn't want, we couldn't give Salvinorin A first because then some of the lingering effects might bleed into the placebo scan. And we used 15 mics per kilogram of Salvinorin A. This ends up being approximately maybe 1 milligram per participant. And so we had 2 different sessions. The first one was we had participants come in and they just practiced the inhalation procedure. And then it was also to assess their tolerance to the drug to make sure they wouldn't move a lot in the scanner, because if people move even more than like 2 millimeters in the scanner, it could totally kill your data. And then the inhalation procedure involved actually exhalation for 5 seconds and then inhalation for 45 seconds, which seems really intense. But when you're doing it through a tiny tube, it's certainly possible. And there was no hold. It's just the hold was happening as they're inhaling for 45 seconds. And this seems to produce the effects that, the desired effects. And then, at least in this first session, we had participants rate the drug strength throughout the trip. So every couple minutes or every minute, we had them rate their drug strength for the first 5 minutes. And then at 10, 15, 20, 25, and 30 minutes, we had them rate their drug strength. And most participants were able to do this. Some, I think, were too off into space, but most of them were able to do it even though they didn't necessarily want to. And then in the second session, we had as I said, the placebo scan followed by a drug scan. And in this situation, we didn't ask participants at all to rate their, we didn't ask them to do anything during the scan other than just lay there and enjoy the ride. And then in both sessions, we had participants wear eye shades, and then they listened to music. And then we also had an experimenter place a hand on their leg. And I actually came into this study about halfway through data collection, and when I found that out, I thought that was a little weird. But believe it or not, a lot of participants actually did find that to be like grounding. They said that that it reminded them that they were in a study at Hopkins, and that they're under the influence of a drug. And then after both sessions, we had retrospective questionnaires, none of which I'll be presenting here. I mean, there's been enough studies at this point, I feel like, that have shown salvia's subjective effects, and frankly, I find them to not be super interesting. So let's look at some brain data. But before that, let me just tell you real quick that so we had only 12 subjects. This is a huge flaw across all psychopharm studies. Funding is really short, and so we only have 12 subjects, which is an extremely low end for an fMRI study. But what I'm going to try to do here is just show you individual data points that across most of the effects that I show you, almost every single subject shows an effect, and in some cases every subject shows the effect that I'll be showing you. So I think it's the best we can do, at least statistically at the moment. And unfortunately, they were all males. These are just the people who qualified for the study and then who could come in. I mean, so maybe this doesn't, I don't know if it does generalize to women, but I'm pretty sure at least what some of these effects do. And there's a pretty wide age range. These were all also hallucinogen experienced. So they had to have had at least 10 lifetime uses of any hallucinogen. They had to have had some exposure to an inhaled hallucinogen, such as like DMT, 5-MeO DMT, or salvia itself. And they had to have smoked something in the last year just so that they could handle whatever they're inhaling to their lungs. And then their salvia uses, actually, some of the participants actually had no exposure to salvia, and then some of them had quite a bit. And then these were all like healthy participants that were assessed by clinicians, and then they were all right-handed, which is something that we do in fMRI studies, because sometimes you can get different lateralizations in left-handed people, which can kill your ability to detect an effect. So this is, rather than telling you what the apparatus is, this is it. So on the left, you can see me holding a little bulb of Salvinorin A. That little white blip, like blip is the Salvinorin A, so it's a really tiny amount. And on the right, sorry, did I say, yeah. So on the right now, you have the stand, which is, there's a stand that the bulb is now attached to, and we have a tube that's attached to that bulb, which goes into the scanner room, because you can't bring metal into a scanner room. And so it's a 690 centimeter tube that goes out into the scanner room that participants would inhale from. And it's got a low coefficient of friction, so we're actually going to do the analysis, but we're hoping that there's not a lot of deposition of the Salvinorin A in the tube. And then you can see that there's two blow torches that we use to light the Salvinorin A to vaporize it. So here's a bunch of the parameters, the fMRI parameters. I just put this up here in case anybody cared. What's important to mention is that both scans, the salvia scan and then the placebo scan, were both 20 minutes long. And we use what's known as a multi-band sequence so that we can sample the brain about once every second. And then again, some pre-processing steps. Again, I just had this up here in case anybody had questions, but I doubt many people do. What is important to mention is that we used what's known as the Shen Atlas. So this is an atlas that parses the brain into 268 different regions or nodes, which results in 35,778 different pairwise connections between these regions. So these are known as edges. And what I like about this atlas is that it's been shown to actually be extremely predictive of certain behaviors. And so it's something that I find really important, even though I'm not going to be showing you too much behavior. So one reason why I wanted to use this atlas. It's also been shown that this atlas can be broken up into 8 different networks, which includes the frontoparietal network, the default mode network, a subcortical network that includes the salience network, and then a medial and a lateral visual areas. And the reason I'm focusing on these is this is where we found our effects. First of all, what is static functional connectivity? So this is the time series of 4 different nodes of the default mode network, and what you can see is that they kind of fluctuate in sync together, suggesting that they're communicating with each other. And so what you can do is you can just basically take a correlation across that time series to get like a measure of connectivity. And so one thing that I've found kind of unreliable in the literature is the way people measure this correlation between different regions. And so sometimes with the default mode network, they'll just pick one region and then just choose to correlate that with all the other regions of the default mode network, or they'll choose to correlate with just one region. So I chose, because I wanted to take an extremely conservative approach, to correlate across every single region of the default mode network with itself, for example, and then just average that correlation. So as you'll see, there's a lot of noise in the Shen atlas. So the default mode network is not a pure default mode network, but essentially this should kill my ability to detect an effect. So if I do see an effect, then we know that it's robust. And so that's just the point I wanted to make to kind of get around this idea that we have a really low N. So first of all, what are some of these subjective effects we can get from Salvinorin A? Well, clearly it works. You can see that in the first couple minutes, people are kind of peaking out. And then at about the 5-minute mark, there's quite a bit of variability in terms of how long it lasts. There was definitely at least one participant that was totally sober by 5 minutes, or at least they claimed they were. But then there were several participants who, even at 15 minutes, were feeling the effects of Salvinorin A still. So this is just showing you that it worked. So this is what, if I look at just every single edgewise, every single pairwise connection between nodes, so every edge in the brain, and then I use an extremely conservative threshold to show what the connections are that exist, this is what you end up seeing. So first of all, the red connections are positive functional connections, whereas the blue edges are negative functional connections. And can anyone guess which one is the salvia brain and which one is the placebo brain? I'm assuming many of you think that it's the top one is salvia, but it's not actually. So here's a bit of a difference that we see with Salvinorin A compared to psilocybin or other classic psychedelics, is that there actually tends to be a decrease across edges, pairwise connections in the brain, although we do see a little bit of an increase in negative functional connections. So this is just looking across the whole brain and not in like a network fashion, which I'll be showing you in just a minute. So, but before I do that, a lot of times with our data, what we want to do is, because when we're dealing with big data, there's a lot of room for error, and so we sometimes just want to convince ourselves that we didn't screw something up in the analysis. So what I'm showing you here is looking at every single edge in the brain of every single subject and plotting the first 10 minutes of the scan compared to the second 10 minutes. So at least in the placebo run, those edges, those functional connectivities in the first 10 minutes shouldn't change too much from the second 10 minutes. And that's exactly what we find in both the placebo condition and the Salvinorin A condition. However, one thing I noticed was that most subjects seem to see a little bit of a decrease in that correlation. Whoops, well, it was supposed to, these things were supposed to cross together. And you'd see actually that the slope of the line on the right is actually less than the one on the left, with the idea being that the first half is not as predictive as the second half on the Salvinorin A scans. And another way to visualize that is plotting every single subject's R value when you're comparing the first half of the scan compared to the second. And what you can see is that Salvinorin A has a much smaller, the Salvinorin A scan has a much smaller correlation between the first half compared to the second half, suggesting that the drug effects maybe are coming on and then they're kind of tapering off. So they're in two different states compared to when they're on placebo, where they're just in one state the whole time. And so every single subject is seeing a decrease in that correlation value. But what's worth mentioning is that there's also a lot more variability in the Salvinorin A scan in terms of that. So some participants are seeing just as strong of a correlation in the first half compared to the second half, but others are seeing, we're not seeing as strong of a correlation, suggesting that maybe there's some participants are having maybe longer lasting effects or shorter lasting effects. So this is the first network that I'm showing you right now. It's a default mode network. So as you can see in the top right corner, that's the Shen atlas's representation of the default mode network. There is a medial prefrontal cortex node. There's also a posterior cingulate node. But there's also some noise, for example, as you can see in the cerebellum, which should be leading to a decrease in my ability to detect an effect. However, when I split my data up into the first 10 minutes compared to the second 10 minutes, you can see that every single subject, that's what every subject is plotted as a point, is seeing a decrease in default mode network connectivity. So what we have on the x-axis is now we have time. On the y-axis we have the average connectivity across the default mode network, and then in the different colors we have our placebo condition and then our Salvinorin A condition. And in the first 10 minutes, every subject sees a decrease in default mode network connectivity, whereas in the second 10 minutes it's a bit all over the place, although if you were to just look at means you would see that there's still a decrease in connectivity. So then the next network, or sorry, one thing, this is actually an analysis I literally put in like last night. So one thing I was curious about was whether or not the people who were still seeing decreases in default mode network connectivity in that second 10 minutes, were they also people who maybe had they had drug effects that were lasting into, let's say, 15, 20 minutes? And so that's what this is actually showing. is that what you can see here is on the x-axis is drug strength, and on the y-axis we're looking at the difference in connectivity between the second half and the first half, with the idea being if there's less of a difference that the drug effects are still persisting through the 20 minutes. And what you end up seeing is that there ends up being, even though it's only 12 subjects, a significant negative correlation such that, yeah, the people who have a greater drug strengths that last into, let's say, 15 and 20 minutes are the same people who are also still seeing default mode network differences in the second 10 minutes of the Salvinorin A scan. So the next thing I'm showing you is the frontoparietal network. So this is like a task positive type network. And what we see here is that, again, Salvinorin A significantly decreases connectivity in this network, but it doesn't really do it in a temporal fashion. It's happening both in the first 10 minutes as well as the second 10 minutes. So contrast that with what was going on with the default mode network. And then lastly, what you can see here is it's the subcortical network, which includes the salience network, but it also has many other subcortical regions. And again, these are significant differences, although if anything, the effect is stronger in the second 10 minutes than the first 10 minutes. So this is kind of like the opposite of what we were seeing with the default mode network. So did all we see was decreases in connectivity? No, we actually did see between these two visual networks, the medial and the lateral visual network, we actually did end up seeing increases in connectivity in both the first 10 minutes and the second 10 minutes. So I don't think these are necessarily, this isn't necessarily a signifier of like maybe complex visual, like visual hallucinations, like the landscapes or whatever that people report, but perhaps it's related to something more rudimentary, given that it's still lasting into the, 10 minutes later, sorry, 20 minutes in. So the next thing, this is where the animations really would have been important, but this is the last measure I wanted to show you. So what I showed you was static functional connectivity, which is just looking at the correlation across the whole time series of two different nodes in the brain. But another way to look at this is looking at how variable are those connections. Two nodes might be talking to each other most of the time, but they might also be doing other things, talking to other people. And so this would give me a high strength of connectivity, but it doesn't tell me how variable that connectivity is, or if another node is talking to someone else. So one way of doing that is instead of taking the correlation across the whole time series, you can take a window, you can just take one window and take the correlation there, and then what you were supposed to see is this window sliding down such that you can generate a time series now of correlations. And then with these correlations, with their powers combined, you can generate a variance term. And this now gives you a measure of how variable is the functional connectivity. And so when I looked at this measure, this is by the way still a super preliminary results. We're trying to look at better ways of doing this. But at least when I looked at across many different kind of time windows, what we can see is that there's actually decreases in dynamics in the default mode network, which I don't know I can't remember if it was a default mode network, but at least one thing that was shown with LSD was that at least across certain nodes, I think, of the default mode network, you'd actually get increases in dynamics. But in this case, across the whole default mode network, if anything, we're seeing decreases in dynamics. And I realize those violin plots are all over the place, but those differences are significant. Then if I look at those two visual networks that were functionally connected to each other, again, we're seeing decreases in dynamics. And so one thing that, Fred, my boss, is a musician, and his kind of analogy to this was that if you're going to become more synced up with somebody else, like if you're going to play music with somebody else, you need to be less variable in what you're doing in order for somebody else to be able to sync up with you. So I think that could be what we're seeing here, is that in order for these two networks to sync up with each other, they need to become less dynamic within their own activity. So in conclusion, what we can say is that inhaled Salvinorin A can be safely administered to subjects in the scanner. I think that was something that one of the things that we can say about this study. And that what was actually kind of surprising to me was that I think every subject that we ran actually reported it being a pleasurable experience. So I think there is something to be said about being in the right state and being in the right context to be able to not necessarily have a bad trip on Salvinorin A. And then overall we saw less connectivity in the brain, which contrasted quite a bit from what we see with classic psychedelics. However, much like classic psychedelics, we do see decreases in connectivity across these canonical resting state networks. And then the most robust effects were in the default mode network. With that being said, a lot of people like to, I kept hearing this yesterday, that the default mode network is associated with the the self. Default network is affected by amphetamine as well. It's also affected by alcohol, alcoholism. So if anything, what we saw is that it just correlated with drug strength, which is a coarse measure of just saying how much do you feel a drug. So I think if anything, when the default network is being hit by these drugs, it's definitely just maybe a more kind of general effect rather than being associated with like self, dissolution or whatever. So the other thing that we ended up finding here is that we did not observe greater connectivity between the default mode network and some of these other networks, like as is observed with ketamine and classic psychedelics. And then we did observe, however, greater connectivity between visual regions. And then last but not least, we observed less dynamic connectivity across the default mode network and those two visual networks. So what was kind of funny is that guy, Eric, the Salvia guy at the beginning, he had an interesting quote. He was also interviewed by Vice at one point, and they were talking about how like bad of a drug it could be, but he actually had this quote where he was like, well, you can actually have a hallucinatory journey in the course of 5 minutes, and it's interesting that we can make our brains do that. If we were allowed to study it further, we'd be able to do some interesting stuff with it beyond getting people high, so I'm trying to fulfill that dream for him. So I'd really like to thank my lab. So this is our lab over there. I'd especially like to thank Fred. Fred was actually one of my friends whenever he was doing his PhD at UC Davis and I was a lab manager. And he's now become my boss, and he entertains all of my old ideas about memory, and he's taught me so many new things, and I haven't even been working with him for a year. Thanks a lot, Fred. And then Roland Griffiths, who many of you all know, he entertains all of Fred and my like weird cognitive ideas. He's been a great boss as well. John Clifton is sitting there 2 seats to the right of Roland. He's been like my partner in crime. He ran every single Salvia subject, and I think he's going off to med school next year. Derek May, who's now, he helped get the study up and running, and he's now a MDMA PTSD therapist out in Colorado. And then, of course, I'd like to thank the brave souls who decided to go into a confined space with loud noises under a potent hallucinogen. Thank you so much to them. And then, of course, our funders. And then y'all should follow me on Twitter. Cool.
Thank you.
Yeah.
All right, guys, we're gonna take questions for 15 minutes for questions. There we go.
I've read that salvia in general has a higher likelihood of adverse experiences. Do you think than say like classic psychedelics like LSD or psilocybin? But do you think that if taken in properly controlled settings, it would have just the same likelihood of positive experiences?
I don't know. I mean, the thing is, our pool was rather biased, because these were all people that had exposure to hallucinogens. Although, with that being said, if you read the Vice article of one of our participants, he actually said that he'd never had a good experience before, and that they were not exactly what he'd call pleasant. I mean, he maybe even referred to it as unpleasant, but he had a good time, and he says it in the article too. So. I don't know. I mean, I think it is a unique drug. It has these weird kind of bodily sensations that could be really uncomfortable for some people. And I mean, I think to compare it also to classic psychedelics, you'd have to compare it to something that could be inhaled and is very rapid acting. So something like 5-MeO DMT or DMT. And I mean, although a lot of people do say that DMT is pleasurable, there's definitely people who have bad trips on DMT. And at least I knew one person who at least said that the closest thing they could compare it to was salvia and how negative it was. So I think if you have anything that's super, super rapid acting and it hits you really, really hard, I mean, I think you increase the likelihood that you could have a bad experience because you don't have time to kind of maybe like integrate what's as your body is changing. Because I don't know if people have as bad of experiences when they take salvia sublingually, and that hasn't been studied super well. But that's my guess is that. Yes, maybe the right context, and if you're in the right state, that I think people could potentially not have bad experiences, but I wouldn't generalize that to everybody. Yeah.
Manoj, thank you, great talk. I especially appreciated the methodological rigor that you used and presented. My question is, what are like the limitations, the big limitations of fMRI studies? unsubstantiated claims that might be made?
There's a lot of them, such as like the default mode network being the self. But if you did notice, I mean, this is extremely like preliminary work, but one thing I tried to refrain from was I'm essentially letting you all interpret those findings. I told you what those networks do, and then I told you what Salvinorin A does to those networks. But I don't want to necessarily, this is what's known as like a reverse inference, is when you read a picture of the brain and you say that this must be going on in the mind without having any kind of behavior to observe. And the field of psychopharmacology has been notorious for this. No names, but you know, they have no behavior, and you can collect behavior, by the way, in the scanner. It's not impossible, and cognitive neuroscientists do it all the time. Obviously, with something like Salvinorin A, like good luck trying to get anybody to do a task with like a button box as they're, you know, off in hyperspace. But you know, I think that you know, that's the thing. So it's like at the moment, I kind of just think of fMRI as being with at least resting state drug effects as just being, let's just make some maps and try to see if these maps can eventually tell us something about what's going on in the mind. But I don't think that, you know, fMRI in general, people have even made the argument beyond drug research that it's told us nothing about cognition. And I don't fully buy that. I think it has told us it's settled certain debates in cognition, but it is the case that people just sometimes get a fancy behavioral effect and just scan it, and I don't think it's a bit vacuous. It doesn't tell us anything. And here I am doing that. But yeah, I think that's a huge limitation of any brain imaging technology. I think we need behavior. And you know, as coming from a behavioral background, I always talked about the brain, but a lot of times when we're reading the brain, what we care about is the effects on behavior. And so I think we need a lot more of those, and that's been, I was actually giving some grief to David Nett. It was really funny. But it was just like, yeah, y'all never measure behavior. And he's like, we're trying to do that now. I was like, okay, cool. But I think that if any of you all, I recommend if any of you are trying to get into this work, become an expert in a certain area of behavior. And then if anybody ever wants to, for example, study perception, the effects of drugs on perception, you'll be the person to go to because you have a background in perception. And unfortunately right now, that's like, I mean, I have a background in episodic memory, but there's actually not been, perception seems like, for example, one of the easiest ways to study hallucinogens, or the first thing that we should be studying, and it really hasn't been done very well. So yeah, lots of limits to fMRI. Sorry.
Thanks for a great talk. So, canonically, when people talk about salvia, the effects of Salvinorin A, they talk about immersive visual hallucinations. I'll just say someone I know has had experiences with salvia that are primarily characterized by very powerful, unusual distortions in spatial and kinesthetic perception, rather than classical visual hallucinations. And I was wondering just sort of what your perspective on the brain underpinnings of that might be, based on your understanding of how Salvinorin works.
I mean, so somebody mentioned grid cells earlier. I feel like that definitely, for example, like, so spatial representation in the brain there seems to be some really specialized cells. Like a lot of times we don't even talk about, when we're talking about like behavior in humans, we talk about kind of global networks or even brain regions, but there does seem to be a lot of real estate in the brain dedicated to spatial representation. So like grid cells and place cells, for example, these individual cells can map out space. And I mean, this is where behavior would come in. If we had a scan where we could measure, people have now kind of figured out a way to get like a grid cell response from fMRI data. It's not obviously at a single cell level, but it's akin to like a grid cell response. That would be something interesting to look at, but no one's done it. I don't know. I mean, people do get these weird kind of proprioceptive effects from salvia. It seemed to be a lot of people would get kind of like certain spinning sensations or like flipping sensations or something. But yeah, again, that's I think that is probably something different about salvia compared to other from classic psychedelics. But yeah, I don't know exactly. You know, I don't want to go beyond my data and say what that necessarily means. And I could potentially, look at the hippocampus, which has place cells, or the entorhinal cortex, which has grid cells, and try to look at those areas specifically and see what happens. But we only have a sample of 12. I'm almost certainly going to find something significant, and I don't buy it. I want to take the most conservative method, and then if something still comes out, those are the effects that we can say that we got from only 12 subjects, whereas all these other drug studies that have come out, I mean, there's been some inconsistencies between the psilocybin and LSD studies, and I don't know if those are true differences in those two drugs in terms of their effects on the brain, but Yeah, it's difficult to say because when you have a really low N, those studies also only had like an N of like 15 or something. So unfortunate, yeah. Whoever, someone, okay.
Hi, thank you so much for your presentation. I especially appreciate you reflecting this on the DMN and self-confident consciousness regarding psychedelics. My question involves your opinion on Robin and Harris's theory of the entropic brain. And in this theory, to put it really simply, the brain's occupying states between chaos and order, and like pop culture, pop science articles describe the brain as regressing back to a baby, even though what's going on is more complicated. And I'd be curious your opinion of this theory and also how the findings of salvia fit into this theory. And also if there's something going on there between perhaps the theory being more applicable to psilocybin and LSD and less applicable to DMT and salvia.
Well, I think it probably, if it applies to psilocybin and LSD, I think it probably does apply to DMT because those are all pharmacologically similar compounds. And it was also actually shown with ketamine, which is kind of strange because ketamine is also an anesthetic and generally entropy decreases under anesthetics. It's worth mentioning David is not on those papers. But so I think it is a bit controversial. I don't know exactly, we were thinking about actually looking at entropy in these data. But I think it's, I'm a bit afraid of entropy because I think people will start to think that, oh, hey, the universe is entropic, therefore this is making us more one with the universe or something when we're under these drugs. But I think it could be something. It's definitely less of I think it's got more content than some other measures, some kind of other nonlinear measures in the brain, like fractalness is something that people look at. But as far as just saying that, oh, there's chaos in the brain when you're underneath the drugs, like I don't think that that's totally right. And I don't think they see entropy across the whole brain. They see it only in very specific nodes that they're kind of cherry picking. And so if you were to if there was total entropy, then we wouldn't have our normal experience, and yet people are still generally able to function under psychedelics. There's certainly impairments in cognition, but there's not full-out entropy either. So I think, it's a novel idea, and I think it's better than the default mode network losing yourself, which I think that is something that maybe he's even moving away from. But yeah, I think that means something that we would love to test here, and I think especially using maybe like machine learning, for example, might be a better way to say that, is there predictiveness in terms of the salvia state from something like entropy, and does it give us more predictive power than like static functional connectivity or dynamic functional connectivity, for example, and that would be one way to, I think, answer that question.
Thank you.
There was oh, cool. Yeah.
Hi. First of all, yes, I also very much appreciated this. I love Salvinorin A. My favorite drug to think about and experience. And it's legal in California, which is where I'm living. Yeah, my question was regarding you had people rate the intensity of the drug experience, I think on a scale of 1 to 10. Did you also collect data regarding, do you know the traditional salvia experience scale?
No.
So it's this 5-6 levels, and basically the traditional effects going up and up until this transcendent otherworldly state where you're completely not in the real world anymore. So you didn't collect any of that? Or the people's subjective interpretations of the numbers that they were rating it as?
Well, at least while they're on the drug, all we collected was drug strength, just to be able to show that there was a drug effect. There were retrospective questionnaires that included, for example, the mystical experience questionnaire. And it's been already shown, too, that there are these mystical effects in the past. As I said, I think we did collect that. I haven't analyzed those data. There's a few other ones that we've added to this, for example, an entity experience or an illusory social event, as I prefer to call it. And yeah, I know we didn't, but we also got their subjective reports as well. They would write what they saw, and I think at least whenever afterwards when we asked people, everybody did seem to have a what was it, transcendent? Is that the term that he was using? Yeah, experience. It seemed to be, anyways. Most people did have a lot of content, although there was definitely some people that actually sort of didn't have a lot of content. One person reported that they felt like they were being like with the researcher's hand on their leg, they felt like they were being held up kind of like a cheerleader, you know, but they didn't actually have much like visual content, maybe like kind of rudimentary visuals. So there was quite a bit of like subjective reporting, but yeah, I like to take that with a grain of salt because there's just so much variability, honestly. Even within individuals, there was a lot of variability, from the first day on the couch compared to in the scanner. And I mean, one thing people tended to report, although I don't think this is probably a significant effect, is that the effects seem to be more intense in the scanner. But yeah, I mean, it's the problem with subjective reporting, as I told you at the beginning, is that you can get weird drugs clustering together that don't probably actually belong together. So I don't know. I mean, I think subjective reporting is one way to measure things. It's not my preferred way of measuring things. Yeah.
Yeah. So then I guess a follow-up to that would be, did you? I guess if you've seen subjective reports, have you noticed any correlations between like the supposed intensity of the experience and what you were seeing?
Well, one thing I'd like to see is to see whether or not, I think, like I said, there was maybe 2 or 3 participants that didn't have a lot of visual content. It'd be great to see if those were also the participants that didn't see large effects in the visual visual networks, the between network connectivity or the within network dynamic connectivity. But no, I haven't gone back to those data. I mean, it's really difficult to kind of go through, and you can really kind of, I think, pick whatever you want sometimes, and just like, there's such a large search space that you can find a significant effect without there actually being one. And so I try to just let, the obvious data speak for itself rather than trying to like look for things that probably don't exist, especially with such a low N.
All right, great. Thank you.
I think there was, okay.
A different flavor than what we've been here. All right, everybody. I just have a few housekeeping